to myself this time.
You get to, yeah. Hey, look, there's nobody here to poke you and mess with your stuff. Oh my goodness. What'll we do? What'll we do?
Oh, that smells so good.
What, bagel?
Yeah.
Yeah.
You're teasing people and you're over there eating a delicious looking pepper as if it were an apple. All right, I can go for that. That's all good.
Why would I waste time and energy to eat something
I mean, for real,
when I don't need?
right? I'm with you. Whoops, whoops, whoops. Go on, do your thing. There we go.
Hello, Peppers.
Yeah, Piper's there. And I expect we'll see Ethan and Piper could be everybody for all I know. It could be her whole house. We don't know. So it could be all the Bensons in one room. Where is this not? Where is the, there we go. Hey, Piper. All right, so we got moving out of our previous unit into our new one. Cells. I love this. This is fun. Ecology is cool. I like ecology, but I really think cellular biology and microbiology and all that.
I'm okay without ecology.
That's fun to me. You're okay without ecology. Yeah, you're like, I've been there and done that. No, I get what you're saying. No, it's everybody has something they like better than something else. Ecology is fun. I like it, but I really like this stuff. This is my stuff. It's exciting. All right, so I think we have everybody in here that's here. Salva, you want a seat? Maybe there's nobody here. You can pick a whole chair.
I like the floor.
Like half the class is out today. All right,
You stole my
the floor is good.
How about that?
Michael likes the floor and the corner where all the dust collects by the vent, and he doesn't seem to care. So, all right.
And I would dust my eyes.
You what?
And I would dust my eyes.
Oh, yeah. Well, that's a good spot for you then. It makes
It's
sense.
really cold though.
It is really cold. I like that. I like that. I'm all for sitting by the vent.
Awesome. What are you doing?
Ozzy,
Trying to move.
do not beg.
Well, there are more dust mites than people.
Come here.
So,
That's
like in
true.
the whole world.
Sit.
So,
Down.
that's crazy.
Nah. Down.
There are more ants than people.
Bro, if enough ants didn't like us.
It'd be terrifying.
All right. So moving on. We're moving on with cells. So we're going to talk today, history, background, cell discovery, how we got to where we are, all that kind of fun stuff. And this is cool piece of history. So first we have the first figure we talk about is Robert Hooke. That's Robert Hooke. He looks rather sullen, but it says behind him, scientist, architect, engineer. Everybody was a polymath, right? Everybody did all things back in the day. If you were educated, you were just educated. You probably spoke a bunch of languages and knew how to play an instrument too, because they all I don't know, maybe he didn't. But anyway, Robert Hooke, 1665. So he viewed cork under a microscope, and the chambers he called cells, just like a jail. Yeah, that's what he's thinking. He's thinking this looks like jail or prison. That's exactly what he's thinking. So when you think of cell, it's the same. Yep, it's the same thing as a jail cell. That's where the idea came from. And so this is 1665. So if you think about the world in 1665 and what was going on, by 1665, we had, colonists over here, right? America's a thing. It's not an independent nation, but it's a thing. So, hey, Ethan. So then the next person that comes along and does something remarkable is, anybody want to try it? Anybody want to try it?
Anton von Leeuwenhoek.
Yeah, so usually English folks are going to say it's Anton van Leeuwenhoek, but I'm pretty sure he would pronounce that Leeuwenhoek. So yeah, because that's one of those interesting W's that becomes an F. Yeah, there you go. So he looked at a drop of water and saw organisms. So you got to figure he's using a microscope. This is 1674. So that lamp that I have over there, that microscope is like 1890s. That's way more advanced than what he's using, clearly. So whatever it is he is using is probably got multiple magnifying lenses, right? You're getting the idea, or maybe a really good one that he's polished and got shaped just right. so that he can see what's in a drop of water. And he would have seen something like this, organisms in a drop of water. So this is the first time human eyes have been laid on microscopic organisms that are actually individual organisms that are living, at least the first time that's been recorded that anybody knows about. All right, and then we get Matthias, this dude, yeah, Matthias Schleiden. And he says, all plants are made of cells. He makes this broadcast statement, this blanket statement. So he's the first person to come out with that. Now check out the microscope he's using. Like that one over there, its light source is a mirror. So you got to bounce the light just right up the tube so that you can see. That's the only source of illumination, right? Schleiden. And so he would have seen something like this, but nowhere near as nice, right? Nowhere near as clear, nowhere near as fancy, probably not as much color. And then we get Theodor Schwann. And he says, well, all animals are made of cells. Now he also invented, this is an aside, but this is what I love about these dudes. Remember I said these dudes are polymaths, right? They come up with all kinds of stuff. So this dude who has made the blanket statement now that all animals are made of cells also invented this rebreather. You know what a rebreather is, right? You know, we talked about him in the last year when the pandemic hit. Oh my goodness, do we all need like scuba diving rebreathers? Okay, but are you ready? You ready? This is his rebreather. You ready? Sure. Okay. This is what this man invented. Yeah. I'm sorry, but that had to be included because, whoa. Yeah, basically you're rebreathing air. You know, you could go in a room with contaminated air or whatever. But I think in particular, the straps on the face.
Directly down his face.
Yeah, no, comfortable. Right over your nose is squashing. I mean, like, look at there. Look at that. Heck yeah. We all want this. Right? I mean, look. I mean, he's legit because I mean, I'm pretty sure I could be wrong, but this looks like him to me. Look in this picture, or maybe it's somebody he knows. I don't know if that's him or not, but if it is him, he puts his money where his mouth is and puts his weird contraption on. But I love this patent for this thing. This is super. These guys, they're great, man. So then the next thing we get is Rudolf Virchow, and he says all cells come from existing cells. Well, now it's 1855. So if we look back here over our timeline, we're going from all the way back here, Luffenhoek, pre-American Revolution, and then it takes a fair amount of time before you get to all plants are made of cells, and the year for that, 1838, and then we get the next year. Well, if you're going to say that, I'm going to say all animals are made of cells. So you're not going to get all the credit for, coming up with these blanket cell statements. Dude, I'm stepping in. And I'm also going to invent some weird business for people to wear. So then we get this guy. All right. And he says all cells come from existing cells. So this is a big advancement because now we're starting to recognize that cells come from cells, which is going to, advance this concept that life comes from life, which is essential because otherwise we're talking about spontaneous generation. So then we put these guys together and we come up with cell theory. Now these are the three main points of cell theory. These are one of these things that's worth memorizing. So it's that all things are made of cells. Cell is the best basic unit of life, and that new cells come from existing cells, which to us seems pretty obvious, but it really was not obvious to anybody that lived before this. So cells, all living things are made of cells. Cells are basic structure. Cells come from cells. Those are three things to definitely commit to memory for like forever and ever. It's one of those forever and ever ones. So now we have different types of cells. So our main categories when we're talking about types of cells are prokaryotes and eukaryotes. So somebody tell me the differences. What's a prokaryote and what's a eukaryote? He's looking. What you got?
Eukaryote doesn't have something separating the nucleus from the cytoplasm, right?
Other way around.
Oh.
Prokaryote. So I'm a eukaryote, you're a eukaryote. We're all eukaryotes. If you remember you're a eukaryote, you got it. All right. Pro meaning before. There you go. Pro coming before. So these are the earliest. Prokaryotes are the earliest forms of cells, and they are not structured as well. They're very small. They're very simple. They're they don't have a nuclear membrane surrounding their genetic information and all the things that you typically find in a nucleus. It's all just kind of floating around, blobbing around. And they have circular DNA and a single haploid chromosome, which means half, not one of those, not a chromosome that looks like this, the X-shaped ones, but just a half, okay? And they divide, when we get to cell division in a couple weeks, they multiply by something called binary fission, which is much simpler than how eukaryotic cells divide. And then basically your prokaryotes are pretty much going to be your archaebacteria, which are your really, really old. Sometimes you just say archaea. It used to be archaebacteria always, so that's kind of what I revert to. But archaea are the really, really old, really primitive, basic life forms, but they tend to live in some very, very extreme conditions. And if you think about early Earth, extreme conditions, right? Your eukaryotes, on the other hand, these are your more complex, right? So eukaryotes have, you were saying it, Michael, they have a membrane surrounding their nuclear material. And they also have all these complex organelles, right? All these little organs that do all these little things. And your similarities are going to be that they both have a membrane on the outside. They both have a cell membrane, or a plasma membrane, as it's sometimes called, which is what we model today, right? And they both have cytoplasm, they have ribosomes, and they have DNA. But everything else is pretty different. So your eukaryotes are your more evolved, your more advanced, your more complex, and your pro coming before, right? These are your ancient stuff. So when we get into cell structure, this is when we start talking about all the different components of cells. Now, if you've ever considered like human anatomy and physiology interesting, where you've got all these different organs that serve all these different functions, cellular physiology and anatomy is equally as complex, or maybe even more so. It's just down to a tiny microscopic. It's wild because, you know, you've got organs that do things, and your organs are made of tissues, and your tissues are made of cells, and your cells have little organelles that do all the same types of things. So cellular organization. So this here is your picture of your plasma membrane. It's one of many, many good graphics that I could find. So what's a plasma membrane? So it's thin, flexible. It's a barrier around the cell. It protects the cell. It's in all cells. Now remember, plant cells have something extra. They have a cell wall, right? Animal cells just have this cell membrane, but everybody has the membrane. Just plant cells have the membrane inside the wall. And then they are composed of what we call the phospholipid bilayer. So here's where we bounce back to chemistry for a minute. So this is a universal component of all cell membranes. Its role is critical because its structural components provide the barrier that marks the boundaries of a cell. Structure is called a lipid bilayer because it's composed of 2 layers of fat cells organized in 2 sheets. So do you remember we talked about micelles a little bit? We talked about like you could buy the micellar water at the store. And basically what it was is it had a surfactant in it that was, it had hydrophobic and hydrophilic ends. This is exactly what you're dealing with here. So each one of these little things here, this is a molecule. One end is hydrophobic, one end is hydrophilic. Do you remember what that means? Hydrophobic, hydrophilic? Yeah. So hydrophilic means, hey, come on. It's not going to repel water, right? It actually would be soluble in water. And then the hydrophobic is not going to be soluble in water. So our lipids, remember that lipids are not soluble in water, right? Lipids are not. Lipids are fats and oils. You can't dissolve an oil. You're going to lick my arm because you're weird. You can't dissolve oil in water. So if you look at the way these are structured, you basically have these, the external surface and the internal surface, and throughout it are going to be these little proteins that make little channels. They're like doors, okay, like gates that open and close. Some of them are open kind of all the time, and certain things have certain sizes and certain chemical makeup pass through. Can you please stop licking me? I love you, but good. Okay, good gracious. And then others are not. Others are going to be controlled by chemicals. They'll open and close based on certain chemical reactions. There are some like sodium and potassium channels that work in your muscle cells that are essential for making your muscles move, and you will be more aware of those because like what's in Gatorade. Gatorade's salty because it contains, yeah, electrolytes, sodium. This is all the stuff we talk about when chemistry, we were talking about electrical ions carrying electrical charges back and forth over membranes. And this is what we mean. So it's also selectively permeable. So what's that mean? Selectively permeable?
I'll let something.
Something's in, something's out, not everything, right? All right. So very complex, even though our simplest prokaryotic cells still have this very complex membrane. So then we have our cytoplasm, and your cytoplasm is your material inside the cell, not including the nucleus. It's the liquid, the gel, the mush that holds everything else. So what was our cytoplasm in our lab today? The starch, yeah. The starch is going to represent our cytoplasm in our lab.
Are organelles considered cytoplasm?
They are, no, they're in the cytoplasm. So Ozzy, no, you're fine. He just hears Jason in the kitchen, so he'll get up and run over there in a minute because he's afraid Jason will drop something. Yeah, well, he's making sure Sava didn't leave any crumbs. Like seriously, he is licking the floor around her. Should there be a single crumb?
It'd be a shame.
Right, it would be. Couldn't leave it behind. So again, your cytoplasm is present in all your cells, although it doesn't contain organelles in your prokaryotic cells, your more basic cells, right? So here is another thing. We talked about plant having a cell wall. So you're going to have a cell membrane in a plant. Absolutely. This is your membrane right here in this diagram. And then a cell wall. So your cell wall in your plants is going to be composed of these guys right here, cellulose microfibers with little pectin strands, interestingly. So this is what you find in algae, fungi, nearly all prokaryotes, and not in animal cells. What do you know about cellulose? Do you remember? We talked about it before.
What about
I know we talked about it last year too. So what is cellulose? What's the common term? What do we, when we are talking about cellulose and we're talking about nutrition, what do we call it? Do you know? Call it fiber. And we can't digest it, right? So that's the roughage when you need more fiber. And people are, oh, you need more fiber in your diet. That's what you're eating is it's plant cell walls. That's all it is. It's what constructs plant cell walls. So you've got this microfibrils in here, which are just tiny, tiny, tiny strands of cellulose. And then pectin, you see how pectin's wrapping around in here? Do you know what pectin's used for? We actually use pectin. Pectin's what makes like jellies, jams. It's the kind of like, okay, you can think of it almost like it's the jelly part. What's the word I want? It's more obvious if you do like say you make chicken stock. Okay, you ever made chicken stock or ever seen chicken stock? What happens when it gets cold?
It's jelly.
Yeah, okay. So that is the animal product. This would be the plant component, basically. That's pectin. And so you'll find it very often if you look at recipes for jellies and stuff, or if you look at ingredients, you'll see pectin on the back. And they extract that out by
Refrigerated gravy after Thanksgiving.
that. Yeah, well, that's the animal component. Yeah, but when we're talking about the plant component, that's pectin. And then you get all these little microfibrils, right? And it's going to be outside your cell membrane. So you've got your cell wall and then your cell membrane inside for plants. And it supplies support for the plant. So when we talk about stimulus and response in just a little bit, have you ever seen what happens when a plant has like sun in one place and it can only get sun in one place? The plant will respond by doing what? What does it do? Yeah, lean towards it. Okay, so basically what's happening in that case, and when we look more at plant cells, we'll see they have that big center vacuole. It's a big, almost like a liquid-filled bubble in the middle of it. And the plant cell can actually control how much liquid is in it, how inflated it is versus how deflated it is. So if my arm is a stem and the sun's over there and I need to get closer to it, there's all kinds of chemical communication within the cell that will tell it, okay, all of the cells on this side need less water in those vacuoles. And all the cells on this side need more. And then what happens then is these become squishier and these become more rigid and the whole plant will bend, right? And that's part of turgor pressure, which is like if you see a limp you've got a flower, you're trying to grow, or a house plant like me, I kill them all. They start looking limp. It's low turgor pressure. It's low pressure inside the center vacuoles in their cells. So it gets really involved. So then your nucleus. All right. So your nucleus, almost all eukaryotic cells, including plants and animals, have a nucleus. Prokaryotes don't have a nucleus. They have a lot of the contents of the nucleus, but remember, it's not inside of a membrane, kind of floating about. There's Alex. Okay. And this is what controls most of your cell processes and where you're going to find your DNA. And then inside your nucleus, so this is your nucleus, right? And then inside your nucleus is your nucleolus, which is this little spot right here. This is where ribosomes are made. And we'll get to what ribosomes do as we keep going. We're going to get pretty intense in the next couple of weeks. And then we have what we call the nuclear envelope. So this is this double membrane layer around the nucleus. So this is what prokaryotes don't have, right? And this allows your material to come in and out of the nucleus, which you're going to need. You're going to need information and material to go in and out of the nucleus. And as we talk about cell development and growth, and when we get talking about sorry, we get talking about how you get from genetic information to actually producing the proteins that are the result of that gene. That's when we talk about stuff getting inside and outside. So organelles mean what? What's it mean?
Any organs.
Yeah, little organs, little tiny organs. Organelle, isn't it cute? It's a little organ. So yeah, it's exactly what they are. Little itty bitty organs. So we have certain types. We have the kind that do storage, cleanup, and support. So this is, we're talking about vacuoles, right? I was just talking about plant cell vacuoles. Are you gonna go? Because I clicked you and clicked you. What's the matter? Hello. Okay, there it goes. That was a lag. Oh, now they're all gonna go. All right, so vacuoles. All right, so vacuoles are spaces or vesicles within the cytoplasm of the cell. So you can think of it like a bubble or a sac inside the cell. And it's got a membrane around it, and they usually have fluid. So they help sequester waste products and maintain fluid balance. So when we're talking about cells in a plant, helping the plant bend towards light, or helping the plant maintain how much fluid, whether that's a limp looking plant or a nice tall standing up plant, that all has to do with fluid balance. But then you have different vacuoles that do different things. So you have a central vacuole. And this is with plants in particular. So this is the one I'm talking about when I say it helps maintain the plant's shape and structure by storing water. Hypotonic. Oh, there's a good one. Anybody want to take a guess at hypotonic? Gotta break that one down. What's hypo?
Very or extra.
It's the hyper.
Oh,
So hypo is
less.
less, right? The opposite, like very little. And what would tonic be? So tonic has to do with tone. You often think of it like with muscle tone is a good example. So for example, some babies are born hypotonic, which means they have low muscle tone. They're floppy babies. That's not a good sign to have a floppy baby. Now, it's not a good sign to have a hypotonic. And there are lots of things that can cause hypotonia. So that's what we're talking about here is a central vacuole helps maintain its shape and structure by storing water. And that would mean it can help it become hyper or hypotonic, really. And then you have a contractile vacuole. So these are going to be found in protist cells. And these actually pump water and they maintain the right concentration within. So these can actually contract and expand, and they can control how much liquid is inside of this particular organism. So these are protists. So protists are, unicellular microscopic organisms. And then food vacuoles. So these are for, they store food for the cell. And then there's this word phagocytosis. Okay, you'll see this in a couple different ways. This is a great word. All right. So phagocytosis is this process of basically something eating something else. So if you've ever seen like a Pac-Man looks where it goes, this is the same idea. So these vacuoles can turn in from a round shape into like a crescent shape, like a moon, and wrap themselves around something they want to contain, and then kind of blend their membranes back together and snag whatever it is. It's pretty cool.
What do cells usually consider food sources?
Well, so it depends on the cell. So if you're talking about little unicellular organisms, they're just going to pick up whatever chemicals they need in the environment, little carbohydrate molecules or whatever. So like when you're talking about parameciums and euglenas and things like that, that's where you'll see this. Now we have cells in our body that are that use the process of phagocytosis as part of our immune system, which is pretty cool. They will just munch things, literally bend right around and swallow it up. Pretty cool. And then why is this thing not clicking today? Ah, look at that. It's like a delay on my click. It's a lag. Oh my gracious. Wow. I wonder why it's doing that. There we go. So lysosomes, here we go. The last one, it's gonna play now. So your lysosome is this little guy here. It's an organelle in your cytoplasm, eukaryotic cells. So only eukaryotic cells. And it contains degradative enzymes enclosed in a membrane. So these break down excess or worn out cell parts, and they can be used to destroy invading viruses and bacteria. So if the cell's damaged beyond repair, these lysosomes help it induce a self-destruct method. And that's really cool. There is a programmed cell death that every cell has. And it's part of its genetic programming that says, when I get to this point, I'm going to self-destruct. And that's to keep your body from having excess cells. This process, this apoptosis process, which is the cell death process, this is part of what happens with cancer. It's the lack of this process. So basically, the cells don't die when they're supposed to die, and they just keep multiplying. And that's how you get tumors and growths like that. That's usually, or can be, part because this process isn't working properly. So these lysosomes, though, they're full of all kinds of, basically it's like poisonous enzymes. And if something gets into the cell that's not supposed to be there, they can sort of throw out their little hand grenades of bad stuff and kill it, or they can kill the whole cell. It's really actually fascinating we're going to do like how cancer works in a couple weeks, and it's pretty neat. So we have organelles that we did that. Support. Sorry. Okay. So support, cytoskeleton. All right. So you have this microscopic network of protein filaments and tubules within your cytoplasm that you can't see. You want to think of cytoplasm as this mushy stuff, like in your model, right? And it is to a degree, but there's also some structure within it. And it's more like, you can think of it almost more like a woven blanket than a structured building. Like a structured building would have really rigid structures. And that's what you're going to see like in a plant cell wall. But think about like, anybody ever knit or crocheted before? Anybody ever tried it? You've done it? Yeah. You've seen like a knit blanket. They're like an Afghan blanket. And you see how it is all held together. There is structure, but the structure is flexible. That's kind of what you're dealing with cytoskeleton. It's fairly flexible, especially with animal cells. Like you can squish them, right? So gives them shape and coherence and helps cells maintain their shape and internal organization. It keeps these parts over here and these parts over here because maybe we don't want them all smacking together all the time. They kind of need to have their own place. And it provides mechanical support and enables the cell to carry out essential functions like division and movement. When we get to division, you'll see certain aspects of the cytoskeleton have very particular functions. So then you've got centrioles. Okay, and this is definitely going to be involved when we get to division. So these are these barrel-shaped organelles. They look kind of like this, right? And they're located in the cytoplasm of animal cells, and they kind of hang out near the nuclear envelope. So these play a role in organizing microtubules that serve as the cell skeletal system. They're kind of like your, what's a good analogy? It's kind of hard to think. Sort of like if you look at a spider web, how it all starts from the middle. They're kind of like that. Everything branches out from here, and these guys kind of organize them all. So they're put to work with both the process of mitosis and meiosis. So these are two types of division that we'll be getting into. And you can't see them until division starts. But when your process of cell division begins, they're visible. They become visible. The rest of the time, they're just sort of in there. But suddenly they become these very visible points, and you'll watch, they'll organize all the rest of these fibers in such a way that it can pull the cell apart to divide it. And we'll really talk about that when we do division in a couple weeks. So some of our other organelles are going to be the ones we call our protein building organelles. So we're going to talk about ribosomes in particular here. So here's our little ribosomes. This is what one looks like. It's like this thingy here would be the ribosome. And this is what's going on inside the ribosome. So it's actually building a protein here from a chain of amino acids off of a strand of RNA. And that's in a couple weeks, we'll get down to detail with that. But this, without ribosomes, you wouldn't exist, because these are the guys that take the code in your DNA and actually build proteins from it. It's almost like a little room, and you got a strand of RNA that's been built from your DNA, and it goes shooting through this little room, and it's like a factory. And when you're inside there, it reads the code as you go along. It pulls. Remember last year when we did the lab? Those of you who were here last year, we did the lab with we came up with words and we made sentences and all that. This is where it all takes place. It's literally this little factory where they read every code that goes by, and they throw down the correct amino acid, and you get the correct amino acids in order, and then you get the correct protein, and it just kind of spits out the other end. So your endoplasmic reticulum, if it would go, what is the deal here? Okay, your endoplasmic reticulum is a network. Hey,
There it is.
oh,
Hi.
I didn't know you would need it over there today. You got it? Drew can probably help you. I knew you were gonna need it in here, but I didn't know anybody was gonna be missing from over there today. Yeah. The plug is behind the couch. Let Drew
Yes.
dig it out for you.
Okay.
And then if you need me to come turn it on, I'll come do it.
Probably.
Yeah, get it set up. I'll come. It'll only take me 2 minutes. So your endoplasmic reticulum, this is a network of these little membranous tubules, these things right here, right? And they are continuous with the nuclear membrane. So you've got your nuclear membrane, and then these guys are kind of stuck on the outside of it, all right? And it usually has ribosomes attached, and these are involved in protein and lipid synthesis, like we were just saying. The ER, which is what we're going to call endoplasmic reticulum, it's classified into 2 functionally distinct forms, smooth and rough, all right? The morphological distinction, that's a fun word. Morphological, anybody want to go for what that is?
Changing.
Study of change.
Yes, you said form. Yeah, basically the form. How structurally is it different? What's the form? So the difference between them is the presence of your protein synthesis particles called your ribosomes, right? That's what we were just talking about. So attached to the outer surface of the rough ER. So your smooth endoplasmic reticulum looks like this right here, and it doesn't have any of the little bumpies on it, whereas the rough has the bumpies, and the bumpies are the ribosomes. So in every one of these is where your DNA is being made into proteins. These guys over here, the smooth ones, lack the ribosomes. That's not where that's going on. So here's your ribosomes. They're your little tiny particles, consist of RNA-associated proteins found in large numbers in the cytoplasm of living cells. Obviously large numbers, because you got to produce that protein. And they have 2 functions. They decode the message and form the peptide bonds. So this is where everything happens. That mRNA comes shooting through and it reads it all. All right, let me go turn on that for Jackie. Give me 2 seconds. I'll be right back. She'll take a second, but she doesn't have a password. Yeah.
Awesome.
Sit. Sit. Wait.
Well, that's interesting. So she can't, You've come back to my chair. She can't run her class at the same time because it's logged in as me and I'm logged in here, so it won't work, the Zoom thing. So we had to do it differently. All right, problem solved for now. Sorry about the delay. All right, so back to organelles. Where was I? If protein builders. Okay, so here we are. If this will go. Golgi apparatus. People like to say this looks like a stack of pancakes. I've heard that said before. Kind of does look like a stack of pancakes. Squash. So this is a complex of vesicles. This is a word we use when we're just like little things. We're just going to say, right? Folded membranes within the cytoplasm of most eukaryotic cells. So these are involved in secretion and intracellular transport. So you've got a lot going on inside this cell where you have to get information or chemicals from one end of the cell to the other end of the cell. And that's where these guys come into practice. So the Golgi apparatus, or sometimes they're going to call it a Golgi complex, functions like a factory in which proteins received from the ER, what's the ER? The endoplasmic reticulum, which is where your ribosomes are. They're making the proteins, right? They're making the proteins and spitting them out. The Golgi apparatus is going to then take those and process and sort them to their eventual destination. So think about that. You got a nuclear membrane with some rough endoplasmic reticulum stuck to it, itty bitty little ribosomes on it. Now what they're actually doing is they're taking a strand of DNA, splitting it in half, creating an mRNA, a messenger RNA strand from the DNA strand, running it through the ribosome, and then the ribosome is taking those little nucleotide bases in sets of 3 codons, coding for amino acids, putting those together in a certain order to create a protein, spitting the protein out, and then it's got to go somewhere. So these dudes they're like the admin assistant. It's their job to organize everything. Okay, you're going here and you're going there and you're going here. So all this stuff's coming out fast and it's happening constantly. It's happening now, like speed of light happening inside your body, right? So, and every living thing. So these guys are constantly working, doing all the sorting. So they may send these proteins to the lysosomes, plasma membrane, or they may secrete it. So it's going to go outside the cell. And in addition, as noted earlier, glycolipids and, oh, that's a fun one, sphingomyelin are synthesized. These are all just different things that are used. Myelin in particular is a substance that surrounds the, if you've seen a neuron before, it has that big long tail. That's what's around it. So basically all those proteins are going to be made in your ribosomes and then they have to get somewhere. And the Golgi apparatus is part of getting them somewhere. So
it just takes it and already knows what it reads it, and then
yeah,
by reading it.
Yeah, it's sort of like the post office.
Yeah.
You know, like you write the letters and send them all to the post office, and then somebody at the post office has to decide where it all needs to go. Well, this pile stays within Georgia, this pile goes to Colorado, and it further sorts it. So then you've got the organelles that are responsible for capturing and releasing energy. So you've got your 2 main types here. You've got your chloroplasts, right? And these are in what kind of cells? Animals have chloroplasts. Do plants have chloroplasts? Where do we find them? Plants, because they have your chlorophyll. That's where they are. So these are the ones that conduct photosynthesis. So this is a diagram of what a chloroplast would look like. And they have these little stacks in here called thylakoids. And you get all the way down inside the thylakoid into this lumen. And this is where your actual reaction is taking place. It's nuts because, you know, you can look at this and go, okay, I can picture that. But we're talking so teeny tiny, right? And everything, chemical reaction tiny, in that little space is where it's happening. So these capture your energy from the sunlight, convert it and store it in ATP molecules and NADPH, which is, we're going to talk more about ATP and NADPH in the next couple weeks. But this is the stuff that, these are the molecules that basically run your energy. Your energy comes from the form of ATP, which is adenosine triphosphate. And that's all your energy is basically in that form. So your other guy down here, whoa, white. I don't know why it hates me today. Just will not click. Okay, again, you're making me nuts. other guy down here is your mitochondria. All right, this is your double membrane bound organelle in most eukaryotic cells. This would be plants and animals. And this is where you're going to generate your ATP that's used as your source of chemical energy. So mitochondria are particularly important. They're also particularly easy to find in a cell. They tend to look like these little oval shaped things with like a zigzag in them. Sometimes people will draw them in a diagram, just like they'll draw a little like a jelly bean with a zigzag on top, and that's your mitochondria. These are the powerhouse of the cell. This is your power plant. In your cells, these are essential. So they are where your ATP is going to be produced. This is where, so if your ATP, okay, here's your infer moment. If ATP is the molecule that stores your energy, okay, then what process is going on inside the mitochondria then? Where do we get our energy from? Not a plant. Plants do photosynthesis. We do cellular respiration is where I'm going. So cellular respiration, meaning, you know, on a large level, you breathe in oxygen, you breathe out carbon dioxide, right? But on the actual chemical reaction level, where that, remember, it's complete inverse of photosynthesis, right? It's backwards, basically. That's going on in here, okay? And there are people that have issues with mitochondria, by the way. People that have mitochondrial diseases, that's really huge. Cause all kinds of problems.
Blood cells go into the mitochondria?
Do the what?
Does like the oxygen from the blood cells get into the mitochondria?
Yeah, it does actually, because all that gets broken down to, you know, the molecular level, and then things flow in and out of all the different organelles. So this was here's your cellular analogy, and I love that somebody used this sign. Props to whoever that is. Huh?
What's up with the office?
Well, because in this case, they're saying, okay, pretend it's a manufacturing plant. Okay, so if it's a factory. So if your cell's a factory, then here's your cell membrane, and this would be like one of your lipid channels in your cell membrane, and it's going to allow, you know, all these particular little elements or molecules to come marching in, and then your ribosomes are over here, and this says manufacturing plant portion. So they're putting in stuff and spitting out fats, lipids, amino acids, and then over here, these guys are packing it up. So these would be your Golgi, right? They're packing this stuff up, sending it out where it needs to go. Dude in the middle is controlling everything. So this would be your DNA, your nucleus, because all this is coded. This is the thing. Your DNA will code for the color of your eyes. It'll code for how tall you're supposed to be, and it'll code to tell your cell, to tell your Golgi apparatus to move protein to a lysosome. Like it's doing all of that, those little teeny tiny things that you never think about. It's coding for absolutely all of it. And then like over here, you got this energy generator. So here would be your mitochondria, and, you know, it's spitting out waste over here. There's always something going on. And then this one, if you were to take this and do an office analogy, all right, everybody here loves The Office. So who is going to be in the middle here? Which character would we slap in the nucleus? We put Michael in the nucleus, we're all screwed. So yeah, but yeah, we put Michael in the nucleus. All right, that's good. So then here is your mitochondria. Here's your work being produced. Who do you think that would be? Yeah, it could be. It actually there are actually people in here, and it's really hard to see. I wanted to find a more blown up version, but I couldn't. But they actually have the characters. I think it's hilarious. Yeah. So this could be anybody who's doing the work, the actual work. So how about, I think this one's easier. Who would your Golgi apparatus be? Who's organizing? Who's really organizing? Who's doing what? Oh, could be. Yeah, I would think Pam too, actually. She takes all the information and kind of moves it where it needs to go. And this ER, this is producing. This is your production part. So I would put the sales guys in here. I'd say, here's Jim and here's Dwight. You know, these are the guys that are actually selling stuff. They're producing stuff. So you can think of analogies like that sometimes, and they're entertaining. Oh, you know, does Toby have to be anywhere? Really, Toby? Maybe the lysosome is Dwight. He's just going to blow it all up. It's just done, man. The what? Probably Creed. There you go. Could be. So cellular transport, how do things get out of the cell into the cell? So here's our bilayer again, our membrane, our phospholipid bilayer. So we talked about how they're selectively permeable. And that's what we were modeling in lab. And they can allow certain molecules or ions to come through. And this is done by two means, either active or passive transport. So active requires energy for things to move. And passive transport is done without cellular energy. So a lot of the passive stuff is going to be what you think of as osmotic, which has to do a lot with balancing the percentage of water on either side of the membrane. So it's important to note that the membrane can also block the passage of materials depending on the conditions inside or outside of the cell. Hello. Okay. So the method used by a cell to pass molecules in or out is determined by the size of the molecule as well as the concentration of the molecules on the inside and outside. Most cell membranes are covered with tiny protein channels, like these little guys here. Ding, 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 ding. And these allow things to move in and out of the cell. And they act like gateways. And we're talking about, it gets down to the molecular level where, you know, little tiny chemical reactions happen to allow things to be attracted or repelled and physically move down. So if a molecule is tiny enough to fit through a special protein channel in the plasma membrane, it will use a form of active transport to move through. If it's too large to fit through the protein, it will have to enter or exit by forming a vesicle. And this is like a little pouch that surrounds the molecule and ushers it through the opening in the cell, just like in this picture right there, where it'll float stuff right out because it makes a little bubble on the outside edge. And this thing will not click. Okay, so here's your passive transport. So diffusion. If you've done chemistry, you might remember we talked about diffusion, about how molecules of a chemical will diffuse through the air. That's how, if somebody sitting over there is wearing perfume and you can smell it over here, it's because they've moved through the air, right? So diffusion here is going to be simple diffusion, which allows tiny little molecules and tiny particles that are nonpolar to cross the plasma membrane. What's polar, nonpolar? Do you remember what the difference was?
Polar is between two, right? And they're it's like opposite attract.
Right. What's water? Is water polar or nonpolar? Water is polar. So what's a lipid then? Nonpolar. So it's nonpolar, and that has to do with whether or not the molecule has a charge at one end or another, right? That will allow these molecules to pass through because it's a lipid bilayer, and the nonpolar heads, the hydrophobic heads, are pointing out when you're dealing with the way the bilayer is constructed. So therefore, if it's nonpolar, it can kind of squish its way right through. Now you have also facilitated diffusion. And this is where you get your larger polar molecules that go across the membrane. So in facilitated diffusion, the molecules can pass both in direction and opposite of the concentration gradient. So here's your picture of diffusion, just squeezing through the little gaps. And facilitated diffusion, you see you have now a carrier protein, which is like, it makes a little channel so they can get in and out. Osmosis. So here's where we deal with our water. Here's our hypertonic and hypotonic again. So osmosis deals with water, usually when we're talking about osmosis. So osmotic pressure. So this has to do always with the balance of water on one side or another. We talk about homeostasis. We talk about wanting this equal amounts of pressure, equal amounts of water, everything balanced, right? So the pressure that must be applied to a solution side to stop fluid movement when a semi-permeable membrane separates a solution from pure water is what we call osmotic pressure. So we said hypotonic a minute ago. So then what's hypertonic? Hypertonic is going to be your higher solute concentration outside. So if you have a cell, all right, and you have a lot of salt water outside the cell, then we're going to say it's a higher solute concentration outside the cell, and we're going to say it's hypertonic. So look right here. Okay, think about a slug, and some poor slug has salt poured upon it. What happens to it? yeah, all the water in its cells is trying to accommodate for the higher concentration of sodium on the outside. So all that water is rushing out of the cells to the outside to try and balance the concentration of whatever is out there, in this case, sodium. All right, so if this is a cell and it's in an environment where just let's use sodium as an example, saline, okay? Sodium and water, sodium chloride and water. So if you've got saline, sodium chloride and water, and you have a really high concentration outside the cell, it's going to do like this guy right here. Because all of the water in the cell, or much of the water, is going to try and move out because it's trying to dilute what's on the outside so that it equals what's on the inside. So that would be hypertonic. Hypotonic is the opposite. So you've got more sodium chloride, for example, inside than outside. So it's bringing more water in to try and balance. So then what happens? It swells up. Okay. And then, come on. Isotonic, here we go. So in isotonic, you got the same concentration of both solids inside and outside. So this is how this cell is supposed to look. So do you know, would you guess what kind of cell it is based on how it looks? Yeah, it's a red blood cell. That's how it's supposed to look. It's happy. Remember we talked, if you did earth science, we talked about isobars, and iso tends to be like the same. So hyper, higher on the outside, hypo, lower on the outside, iso, the same. Here's a good one, cytolysis. Ooh, anybody want to try and guess what cytolysis means?
Cytolysis?
Mm-hmm.
Is it when the cell goes and gets out?
Yep. So anytime you see lysis, you'll see this a lot or very similar. It means like to cut, to break, to split. So this breaks open and everything comes pouring out, right? So that would be cytolysis. So this would happen if it got way hypotonic, right? It could go pop, and that's what you get. So that's, you know, the poor slugs. So aquaporins, aquaporins are integral membrane proteins that serve as channels in transfer of water, and in some cases, small solutes across a membrane. So it would look kind of like this. Ding, ding, ding. You have, here's your phospholipid bilayer again, right? Hydrophobic, hydrophobic, hydrophilic in the middle, protein, and notice there's a charge. All in the end going to come down to electricity. At the very, very, very end of it, we're dealing with where the electrons are. Yeah, hydrophobic.
Scared of water?
Yeah.
Okay.
Like lipids are hydrophobic. They're nonpolar. Transport proteins. These are actually really fun. They do all kinds of crazy stuff. So transport proteins transport substances across membranes. Now, these will exist in more than just cell membranes. You'll see them later in other things, but for the purpose of cell membranes, they're found within the membrane, they form a channel, and they have a carrying mechanism. So these are things that have like an actual gate, like a physical gate. And that gate is going to function, again, it's going to come down to chemistry and then electricity at the very end. It's always going to come down to electricity. They will open and close. They will open one end and close another. Think about like if you've ever seen a lock function for moving you ever seen a lock function for moving a boat from one elevation to another? Similar kind of stuff. So they act like a pore in the membrane, let water molecules or small ions through quickly. These are the gated ones. They open a gate and allow a molecule to pass through the membrane. And then you have carrier ones. So these are specific for specific ions. So they're going to be looking, these guys are looking only for specific ions. They won't move just anything. They move the one thing they're supposed to move, or the two things they're supposed to move. So they're very specific. So this is just sort of a graphic to show you. So here's a channel protein. Stuff just flows in and out, simple diffusion, or it could squish in and out in between the bilayer if it's small enough. Facilitated, and then you've got carrier, and then some of these are going to require energy, and those are going to be your active ones. So passive here, the passive transport is all this stuff over here that doesn't need energy, and then your active transport's over here. This is the stuff that does need energy. So homeostasis, this is what we're talking about with hypotonic, hypertonic, trying to get everything happy. So unicellular organisms, these little dudes here, amoeba, this is amoeba, right? Paramecium and a euglena. These are like the three that everybody looks at under microscopes. So in terms of their numbers, unicellular organisms dominate life on Earth, way more than the rest of us. And they include both prokaryotes and eukaryotes. Prokaryotes, especially bacteria, are remarkably adaptable and live almost everywhere in soil, leaves, the ocean, in the air, and even within and on your body, all over you, inside you, everywhere. So eukaryotic algae are found in oceans, lakes, streams around the world. Yeasts or unicellular fungi are also widespread. And yeasts play a role in breaking down complex nutrients, which makes them available for other organisms. So yeasts, you've ever had bread, you've had yeast, right? And that's a fungus. And they're unicellular. But when they break down nutrients, they produce gas, and that's how we get fluffy bread and things like that. So this dude here, he's going to show us our organization for life, basically. So, we start with an atom, and of course, beyond the atom, you've got subatomic particles, and then beyond that, you have even smaller things. You get to the quarks and all those fun things, right? So you have atoms together, make your molecules. Molecules are going to compose your cells. This is the part for you to remember, that your cells make up tissues, your tissues make up organs, your organs make up systems, and those make up organisms. So when we talked about cancer, if you're down at this level, the cell level, like cells make a tissue, okay? So all the like cells make the same kind of tissue, and those different kinds of tissues can compose an organ. So in this case, your stomach has several different types of tissue in it, but each of those types of tissues is made of all the same kind of cells. So when you're talking about cancer and you have apoptosis not functioning properly, cell death not functioning properly, you get all these cells, say it is stomach lining, okay? Down in here with this business, your very, very inner stomach lining. We're talking about that epithelial layer, all right? Those cells in particular, if they're the cells that stop dividing and stop dying like they're supposed to, and they start dividing uncontrollably, then if you have cancer of this particular type of cell, when it spreads to other parts of your body, when you've heard about, the cancer metastasized, like he had this kind of cancer and now it's here, it's gone here, it's moved there. If say your cancer is, like, for example, my uncle had kidney cancer. When it got to his bones, if you were to pull those cells out, those are kidney cells in the wrong place, because now they have grown uncontrollably, they won't die, and they're moving everywhere else. So that's what happens when it metastasizes, it flows everywhere. So those cells will get into other organ systems where they don't belong, and that causes major problems. But if you remember this, there's also Venn diagrams, but I like the dude with the fig leaf better. And I made it so fig leaf moves real fast. Real fast. What? So he didn't have a fig leaf. I added it for your sake. I didn't have to add it. I added it for your sake. So yeah. Yep. If you just remember that cells make up tissues, tissues make up organs, organs make up systems, and that makes a complete organism, then you'll be good to go. So these are from your homework, or your notes, sorry. If you have that in there, you may want to look that over and see if you got them right. So yes, your prokaryotic cells are going to be much smaller than your eukaryotic cells. They're going to lack your nuclei, and an example would be bacteria. And examples of eukaryotic cells are pretty much everything else, right? And there are bacteria that are eukaryotic as well. And then specialized cell parts. So cells that line the upper air passages in human cilia, that's what we call them, the little hair-like cells, right? And then their job would be to sweep mucus, debris, bacteria out of lungs. And then here's your pine pollen grain. And it has little wings on it. And it enables the pollen grains to float in the slightest breeze. So when they land in your cilia, you're going to make, that's what your mucus is there, to snag them, right? So then it's the cilia's job to kind of sweep them out of the way, right? And pollen is one of the things that people say doesn't usually, pine pollen in particular, doesn't cause as many allergies as other stuff, because the grains are actually bigger than like oak, and some of the other ones are actually bigger grains. But it's the stuff that when you go outside, I swear you can feel it in your eyes. You know, when the pine trees are like really dumping pollen everywhere, I do. I walk out and I'm like, oh, it feels like sand. Like it's so thick. And you see it on the ground, makes that big yellow blanket. That's that stuff. Yeah. So there's this week. So we're going to be moving on, getting even pickier about what happens inside of cells over the next couple weeks. And we'll be talking about eventually we're going to get to how the proteins are made from the DNA, like really in particular. It's really neat. This stuff gets neat. It's fun. I like it. I think it's more entertaining. So we've got some time. So let's look ahead to this week's homework since we have some time. Sometimes we're going to have time, and I would love to give you the heads up. So I'm going to go to, if it will let me do it without opening up everybody's business. Sometimes it does. Come on. Hello. Okay. Oh, yeah, we did classification with the other group. We'll do that next week too, when we start talking about classifying life. We looked at some fun stuff today. It's kind of cool. That's going to be in my way. Get down there. So this week's homework, if I can make it open up without showing me things I don't want it to show me. Let's see. Come down here. All right, lesson 5, homework. Is this going to be yours or the other teams? I can't tell. There we go. So organization of life Venn diagram. It's a file for download. So let's have a look. Go on. Okay, there it goes. So your homework this week is to take this right here. We were just talking about this, right? Cells, organs, tissues. Your job is to do this as a Venn diagram. So what would you put? I'll make it so I can write on this if I can. Yep, this is just a different kind of Venn diagram. This is the kind that's the circle inside the circle instead of the circles all touching each other circles. A little different.
So what's the middle one, the one they have in common?
Yeah, so the middle one would be the one they have in common. So, you know, we can actually go back over here where it'll let me write on the screen. Let's do this. Okay. So if I put in, so what's going to be at the very center of our Venn diagram? Say it louder. Atoms. Okay, so atoms are going to be at the very center of our Venn diagram, right? Okay. And then what's going to be one step bigger? Molecules is right. So I'm going to send this one to the back. And I'm going to make him a little bit lighter in color too. We'll make it neat as we go. Why not? Right here. So molecules is next. I can't see what I wrote because it went weird. I knew I did that wrong. I need to get rid of atoms, which I can't see what I'm typing here. Okay, so there we got that. Ding ding, sort of. I'm gonna have to make these, you know what? Let me make these so they go. All these things are in my way. There we go. All right, so then what's gonna come after molecules? Cells. This is not clean nicely. Okay, so cells will be next. And we're going to make cells go, we're going to make ours a little diagonal here so that it fits better on this page.
The homework doesn't have atom or molecule on it.
Okay.
Just ordering.
Yeah, so we're letting it go. We're going back even further. We're getting them all. So then we get cells. It should be even easier than the homework, right? All right, and then what's going to come after cells? Tissues. You know what? I want it to be a lighter color though. I'm picky. I like it to be little arrow. There you go. So we get tissues. That's next. Make it bigger. Send it to the back. See, this should be easy. I should be done with it by the time we're done here. Tissues. What comes after tissues? Organs, right. I'll change your colors in a minute. Say what?
The most important step.
Oh, yeah. I have to be picky. Organs comes next. All right. And then what's going to come after organs? Organ systems. That's right.
Did you say?
No. Do I live in Oregon? No. Systems. And then maybe we can go and do an example of each of these. Send it to the back. And then what's our last one? Organisms, right? Not even gonna I'm running out of room, haven't I? That's okay. I can scooch it back still. Do this. Go back over here. Organisms is right. But I can't write it. But you know what's going in there. So what is an example of an atom that might go into a molecule? Anybody want to throw one out there? Hydrogen could go. How about how about something that has more than one element? What's a good? Well, okay, so now we're going to go from hydrogen, and then what was next?
You said no, you were still asking for how to have more than one.
I'm at molecule.
Water.
Sorry, water. Okay. Now, what's a type of cell?
Water.
Where's the type of cell? How about a type of cell in your body?
Red blood cell.
Red blood cells, one. They're a little harder to deal with in this example because they don't make so much of a tissue.
White blood cell.
Same thing.
Skin cell.
Skin cell. Okay. So we're gonna have in particular like let's do an epidermal cell. Yep. So this is gonna be the skin cells on the very outside. Okay. And then I'm gonna shrink this a little bit so we can see a little more. So then what tissue do these cells make? You just said it. Skin. So that is going to be your tissue. In this case, it's also going to be your organ. Yeah, because we kind of call it the same thing, right? Technically, you can have, there are different kinds of skin, when you think about it, but, and then your organ system, does anybody know where your skin's going to fall? What organ system is your skin? Do you know? Integumentary. There you go. And then this last one should say organism, but it doesn't say it yet because I couldn't reach the word. And we'll just say me, right? So that should be an easy one for homework. Y'all should be able to do that pretty easily. And I think y'all did well on the test. I don't think there's anything to like really look over on the test that I'm going, okay, we need to not really do this. Y'all did fine.
I put in C4 and it said it was wrong.
Oh, sea floor. Oh yeah, but it wasn't. That wasn't on this last test.
Yeah, it was.
It was.
Yeah, it was.
Yeah.
Was it? Oh yeah, it was. It was. Okay, I was thinking about the other guys.
I think it said mine was on too, but I said benthic zone instead of. bottom.
Okay. Yeah, those are the ones I can go back and double check. I'll go back and double check that one too.
I think it said water requirements.
I think it was looking for
It was looking for ocean floor.
Yeah.
bottom of the ocean, bottom of the sea.
Okay, I'll go back and double check them all. I gave them all a quick once over this morning at 8 a.m. So I'll go back and do them again and make sure they're all accurate. Okay, yeah, so your homework should look something like this. Not too hard. I can't make it so that you can see it any better. I'm trying. It doesn't like me, but you get the idea. There we go. Sorta. Sorta. So there you go. You can do that. It's easy. Fill that in. What else? Do you guys have questions about lab? We're wrapping up just a little early today, so I want to make sure that
Do you want
we've ever covered
you
up.
want us to write On the homework, do you want us to write cells, tissue, organ system, or do you want us to write
Mhm.
a cell or tissue?
No, do exactly like you just said before. You got the idea right. And then, I mean, you could give an example if you wanted, but you don't have to. So next week, just so we're looking ahead, let's see.
I won't be here in 2 weeks.
You won't be here in 2 weeks. Where are you going?
Why don't we?
You gotta go have fun somewhere, huh? Yeah. So just so you know, we will be, what now?
I've been to Jackson. Yeah, I've been to Jackson Hole. It's a city
Jackson
of-
Hole. So we're moving in. Oh, and your Q2 stuff. If not next week, the week after, you'll get your Q2 packet. It's almost done. So you'll be moving next week into, that's your homework one. That's not the one I meant to click. Here we go. So we're going to talk specifically about photosynthesis. And we're going to be doing, I think the lab is the plant pigment one. Yeah, we'll be doing some plant pigment stuff. That should be fun. And the lab is going to be a no write-up lab. Yay. But we'll start talking about, here's where we're starting. Remember I said we were talking more about ATP. We're going to really get into ATP in the next couple of lessons. And so we'll be talking about that, and we'll be talking about photosynthesis and respiration. And we're going to get pretty down and dirty in the next couple lessons when you get down to the chemical reaction level, which it's just a little in the weeds. So what I ask you to do is kind of keep in mind these next couple weeks. It's really easy in biology when you start talking about the tiny stuff that's going on. It's really easy to forget the bigger picture and forget like even where you are. Like, am I in a plant? Am I in an animal? Am I in a cell? What's going on? So try and keep your bigger picture in mind as you go. Oh, and there's a couple of cardinals if you want to. Yep, he just kicked her off of there, like did not care. So I'm gonna let you go a little early. As long as y'all are quiet and you don't bug them too much, you should be good to go. If you have cleaned up your lab, awesome. But if you still have a lab over there that needs cleaning up, if you can make sure all your starch is in one container, all your iodine solutions is in the other, and then the empty containers are in the bin back there, it would help me greatly. And y'all should be good to go. Is your puppies? All right, guys. I'm gonna let y'all go.
Thank
A
you.
little early today. We're good. Okay, that's gone.